Myocardial infarction or heart attack is a pressing problem in developing countries which kills more individuals than all cancer combined. A single attack destroys about 1 billion cardiomyocytes, accounting to about 25 to 30% of an adult heart. Then the person sluggishly drags along the life with a part dead heart, which then progressively leads to functional loss of other vital organs and then leading to a high mortality rate. There are drugs which are prescribed depending upon the severity of the disorder, but the gold standard treatment till date remains to be organ transplantation, where the donors are way limited, and then that involves a high risk of immune rejection. And this is exactly where my research comes in, where the idea is to design and build a cardiac patch which has the capacity to beat independently so that we can patch up a broken heart. And in order to do so, I divided my thesis into two aspects. First was to increase the production of cardiomyocytes to a very high number, and to do that we combined engineering principles and biology together to give it a whole bio-inspired tissue engineering approach, wherein stem cells were used to mimic a developing embryo and then differentiating that into cardiac cells solely by cell-cell interaction rather than cell matrix interaction. And to do this, we formed these three-dimensional aggregates of cells using 3D printing technology, which enhance the production of cardiac cells to a high number, ensuring the precise control of parameters like the cell number, the cell density, the geometry and the shape of this embryo without use of any other chemical stimulators. And after this was done, I moved on to the next part of my project, which was to build a cardiac patch. And to do that, we wanted to use scaffolds. Now, scaffolds can be natural, can be synthetic, but we wanted to use a natural source of scaffold. We couldn't get hold of human heart-based scaffold because it is ethically questionable, so we rather went on to using porcine-based scaffolds. So these scaffolds were then chemically treated to make them immunologically inert, and then put the cardiac cells produced onto it and then stack them up to a 1 centimeter tall cardiac patch, which is now ready to be implanted into human heart. So this is definitely a long-term project which not only requires proof of concept in laboratory scale but also other collaboration by international agencies so that we can move the research from bench to bedside. So if I were to sum up my thesis in one sentence, it would be clubbing engineering and biology together so that we can fix problems like this once and for all rather than just slowing or managing it down.